Hello everyone, myself Dr. Parth Goswami, and today I am going to teach you about microbial carcinogenesis, means which are the microbe that can cause cancer. So the microbe that can cause cancer could be of virus, bacteria, fungus and parasite. Among that virus that causes the cancer is known by the name oncogenic virus. So virus that cause the cancer or tumor, it could be benign tumor or malignant tumor, is called as an oncogenic virus, right? All right. So first of all, we will discuss about the oncogenic virus that can cause cancer. So it could be of two variety, oncogenic DNA virus or oncogenic RNA virus, right? So which are the example of virus that contain the DNA, oncogenic DNA virus? So it could be HPV, Epstein-Barr virus, hepatitis B virus or human herpes virus 8. The example of RNA virus include HTLV1 or it could be hepatitis C virus. So first let's discuss about the HTLV1 human T cell leukemia virus. So this particular virus, HTLV virus, having the tropism for CD4 lymphocyte, and it can lead to development of adult T cell leukemia or lymphoma. So the tumor produced by HTLV virus is adult T cell leukemia lymphoma, and according to that the name is given to the virus. But friend, interesting fact is that it is having the long latent period of 20 to 40 year for development of adult T cell leukemia or lymphoma. And this particular lymphoma is endemic in a few area of Japan. Now you might have questioned which could be the mode of transmission of HTLV virus. So it can be spread by sexual contact, sexual intercourse, then it can be sprayed by blood products, or it can be sprayed by breastfeeding. Now let's understand how HTLV causes the cancer. Pathogenesis of HTLV induced neoplasm. So friends, remember that HTLV virus is a retrovirus. It having the gag-pol and envelope gene, but additionally they contain the two main gene that is responsible for causing the cancer. The one is TAX gene and second one is HBZ gene. Among that HBZ gene is a transcription factor for the virus, and TAX gene will lead to production of TAX protein, and this TAX protein leads to a viral replication and neoplastic transformation of CD helper T lymphocyte. Now friends, one interesting fact is that the HTLV induced TAX protein, this produced TAX protein can inactivate the P53 gene. And guys, P53 gene is guardian of genome. It is one of the tumor suppressor genes. So if it is inactivated, there is an increased mutation and there will be genomic instability, which will lead to monoclonal neoplastic proliferation of CD4 T lymphocyte. It can lead to monoclonal neoplastic proliferation of CD4 T cell, and so it can lead to cancer of T cell, that is human T cell leukemia virus. Now let's understand about the human papilloma virus, HPV virus. So there are more than 70 genetic different human papilloma virus is identified, but basically HPV virus, it's a RNA virus. So basically it can be divided into two variety: low oncogenic risk or high oncogenic risk HPV, right? It's a DNA virus, not RNA. All right, so now we are seeing the example of DNA viruses, right? Now friends, low oncogenic risk potential HPV will not integrate with our DNA and it will remain free, which is known by the name episomal form. This free form doesn't cause cancer. It can lead to benign tumor. It can be divided into two variety, HPV 1247, which can lead to development of benign squamous papilloma, or traditionally known by the name wart, right? And other type of low oncogenic risk HPV, like that of HPV 6 and 11, it can lead to development of condyloma acuminata, which is known by the name genital wart, right? It is especially seen in vulva, in penis, and in perianal area. It can also lead to development of laryngeal papilloma. So that is about the low oncogenic risk HPV, which will lead to benign tumor. Now let's discuss something about high oncogenic risk HPV type, that is HPV 16 and 18, which are a high oncogenic risk potential. So friends, these particular virus will integrated with our host genome. They will not remain free like that of low risk HPV, right? They are integrated so that with integration of our DNA, the malignant transformation of our cell can occur, and that's why they will lead to malignancy, particularly SCC of cervix and anogenital region. It will lead to squamous cell carcinoma, and oropharyngeal cancer also can be developed in the tonsil. So that is about the high oncogenic risk HPV that will lead to cancer by integration. All right. Now let's understand the pathogenesis of HPV induced cancer, that is caused by integration with our genome. So high risk HPV integrated with our genome so that they will overexpress two particular viral gene which is responsible for cancer development. One is E6 and second one is E7. Both will lead to production of E6 and E7 protein. Now first of all, let's understand how HPV E6 gene, human papilloma virus E6 gene causing the cancer. So the one way is that they will inactivate the P53 gene. The P53 is a guardian of genome. It's a tumor suppressor gene. We already know that. The P53 gene provides the genomic stability to our gene structure. So it's a tumor suppressor gene and it's a guardian of genome. So if it is inactivated, the cancer can develop in the body. The second way of function of E6 gene is increased telomerase reverse transcriptase. There is an increased telomerase reverse transcriptase activity so that cell replicative senescence can be prevented. I want to say that by activation of this enzyme, the telomere will not get short in the each cell division. Normally telomere shorten with each cell division, but here it is not shortened, and so the cell will continuously proliferate, and so the cell will become immortal. The cell will not die, and so they will continuously proliferate and they cause cancer. Now let's understand how HPV E7 gene causes the cancer. So the one way is that they will inactivate the RB gene. RB gene is one of the tumor suppressor gene which in combination with ETF prevent the cell cycle movement, right? It is a tumor suppressor gene. So if RB gene is inactivated, the inhibitory effect of RB gene is lost and E2F will be free to do the cell cycle movement. So RB gene E2F combination will be inhibited, right? So that cell will proliferate continuously. The second way is that HPV E7 gene, can activate cyclin D and cyclin dependent kinase 4. They can activate cyclin E and A as well. So all these are cell cycle movement causing factors. They will continuously lead to cell cycle remain activated because of upregulation of it, and they will inhibit. The third way is that they will inhibit this E7 gene inhibit cell cycle regulator like that of P21 and P27. So the cell cycle will not regulated and cell cycle remain permanently activated, so cell continuously proliferated and causing the cancer. All right. So you can easily understand that both HPV E6 and E7 will provide genomic instability. They will not stabilize our genome. The genomic instability occurs if they are overexpressed. They will activate the cell cycle. They will immortalize the cell by inhibition of replicative senescence, right? They will increase the cell proliferation. So because of all these factor, cancer can occur by HPV high risk type. All right. Now let's understand about the Epstein-Barr virus, EBV virus. It is a human herpes virus family, and obviously it's a DNA virus. It can infect the B lymphocyte. They have the tropism for B lymphocyte, so they will commonly lead to B cell cancer, B lymphocyte cancer. The cancer that can produce by EBV virus is, it can lead to Burkitt lymphoma, that is particularly of African type. Then they can cause B cell lymphoma, especially those who are immunocompromised. Third cancer, that they can lead to development of certain forms of Hodgkin lymphoma. Nasopharyngeal carcinoma can occur by Epstein-Barr virus. Epstein-Barr virus can lead to development of gastric carcinoma, particularly gastric adenocarcinoma. They can lead to development of natural killer cell lymphoma like that of T cell virus, and rarely they can cause sarcoma. Alright. Now let's understand the pathogenesis of Epstein-Barr virus, how they cause the cancer. So we can divide the pathogenesis of Epstein-Barr virus into a three type. Pathogenesis can be discussed in three type. One is pathogenesis by latent infection. Second one is pathogenesis of Burkitt lymphoma. And third one is pathogenesis of nasopharyngeal carcinoma. Nasopharyngeal carcinoma. So let's discuss first latent infection induced cancer, pathogenesis of latent infection. So see friends, Epstein-Barr virus can remain latent inside the B lymphocyte, and they are having the three important gene: latent membrane protein 1, that is LMP1. Third one is Epstein-Barr nuclear antigen 2, and third one is viral cytokine 10. And viral cytokine 10, because of all these three products, two gene and one cytokine, the cell will become immortal, and so they will continually proliferate and will lead to B cell cancer, right? And particularly if we understand in the detail, then latent membrane protein 1, LMP1, will act as an oncogene. If they act as an oncogene, they can activate the NFKB and the JAK-STAT signal transduction pathway, and so that they can proliferate the cell continuously by activation of NFKB and JAK-STAT pathway. The second thing is that they can activate anti-apoptotic factor, particularly BCL2, which is an important anti-apoptotic gene. So if they activate this gene, apoptosis will be prevented, and so the infected B lymphocyte will not get removed by apoptosis, and so the cell will continuously proliferate. Right? Alright. Now, Epstein-Barr virus nuclear antigen 2 gene will lead to stimulation of the transcription of cyclin D. They will lead to transcription of cyclin D and SRC family of proto-oncogene. So because of that, cell will continuously proliferate. We know very well that cyclin D is a cell cycle activator. So the cell will continuously proliferate by doing the cell cycle movement. Viral cytokine 10, VIL10, will lead to inhibition of macrophage function, so that infected cell will not get removed. Alright. Now pathogenesis of endemic Burkitt lymphoma, African type of Burkitt lymphoma. So here the T suppressor cell will not work. If the T suppressing cell is not work, then B cell will continuously proliferate. The suppressive effect of T cell is lost. Alright. The second thing is that, in this particular pathogenesis, there will be chromosomal translocation of 814. The portion of 8 gene translocate to 14, so that they will activate the C-Myc oncogene. CMYC mycogene is activated. So all these two factors will lead to development of Burkitt lymphoma. So that is the pathogenesis of endemic Burkitt lymphoma. Alright. Now let's see the pathogenesis of nasopharyngeal carcinoma by HBV virus, that is of T cell variety cancer. So here obviously LMP expression is increased, so it will stimulate NFKB JAK-STAT signal transduction pathway. They will stimulate NFKB pathway. But additionally, here there is an overexpression of vascular endothelial growth factor, fibroblast growth factor 2, then matrix metalloproteinase 9, and COX2. So because of overexpression of all that, it will favor the oncogenesis. Alright. Now let's understand something about hepatitis B and hepatitis C virus. So hepatitis B virus is DNA virus and HCV is RNA virus. Both these virus, HBV and HCV, can lead to development of chronic liver inflammation, which is known by the name chronic hepatitis. And from chronic hepatitis, patient can develop cirrhosis of liver, because of which there will be development of primary hepatocellular carcinoma. So in this way they can cause cancer. Now let's understand about the HBV hepatitis B virus. So it is having the one important gene, that is HBX gene, and it can cause a cancer in a four way. The one way is that it will increase the transcription factors. It will increase transcription factors. Second thing is that it will increase the signal transduction. Third thing is that it can inactivate the p53 gene, which is an important tumor suppressor gene and the guardian of genome, right? It will inactivate p53 and it will integrate with our host genome. So in this four way, HBV virus can cause hepatocellular carcinoma. Alright. Now hepatitis C virus. It contains the HCV core protein, right? And they will lead to activation of growth promoting signal transduction pathway. Activate growth promoting signal transduction pathway, so that they can lead to continuous cell proliferation and hepatocellular carcinoma, and the lymphocytic lymphoma can be developed. Lymphocytic lymphoma and the hepatocellular carcinoma, the cancer caused by HCV virus. Now let's understand about the human herpes virus 8, HHV8. It can lead to development of three type of neoplasm. One is Kaposi sarcoma, that is the most common and associated with AIDS. It's a vascular tumor. Second one is primary effusion lymphoma. And third one is multicentric Castleman disease. Primary effusion lymphoma and the multicentric Castleman disease is a rare. Common one is Kaposi sarcoma by HHV8. Now let's understand the pathogenesis of HHV8 virus, human herpes virus type 8, how it lead to development of cancer. So one is that they will inactivate the p53 gene, which is a guardian of genome. Second one is that they will inactivate the RB gene, tumor suppressor gene. And third one is that they will downregulate the MHC class 1 molecule. Downregulate MHC class 1, and we know very well that cytotoxic T lymphocyte are MHC1 restricted. So MHC1, if downregulated, cytotoxic T lymphocyte cannot work and they cannot remove the infected cell by HHV8, and in this way they can cause cancer. Alright, now let's understand about the Helicobacter pylori bacteria. So it can lead to development of gastric adenocarcinoma or development of gastric melt lymphoma, which is a B cell lymphoma, gastric meltoma, right? Mucosa associated lymphoid tissue lymphoma. It's a B cell lymphoma, and here the translocation is of T11 to 18. Alright, now let's understand the pathogenesis of gastric adenocarcinoma by H. pylori. So whenever the patient is having H. pylori infection, there will be chronic inflammation of gastric mucosa. Chronic gastritis can occur, which subsequently will lead to development of gastric atrophy. From gastric atrophy, there can be development of metaplasia, intestinal type of metaplasia particularly, from which dysplasia can occur, and ultimately gastric adenocarcinoma can arise. Alright. Second thing is that important fact is that H. pylori is having one gene, that is cytotoxic associated A gene, CAG-A gene, and this particular gene penetrate a gastric epithelial cell. It will penetrate a gastric epithelial cell, which can initiate a signal. So growth factor will continuously get stimulated, which will initiate a signal for growth factor, and in this way they can cause cancer. Alright, aflatoxin B1 fungus. Now let's discuss about the fungus. So one important fungus that can cause hepatocellular carcinoma is aflatoxin B1. Parasite. So common parasite that can lead to cancer development is Cystosoma and Clonochis sinensis. The Cystosoma hematobium can lead to development of urinary bladder cancer, particularly squamous cell carcinoma of the bladder. And Clonochis sinensis, this parasite can cause chronic inflammation of bile duct. chronic inflammation of bile duct, so that because of chronic inflammation of bile duct, adenocarcinoma of the bile duct can develop, which is known by the name cholangiocarcinoma. All right. So this was all about the microbial carcinogenesis. Thanks for watching till the end and hope you have enjoyed this video. If you have any question or suggestion, then you can ask in the comment section. Thank you very much, and I will be right back with a new video. Till then, take care and bye bye.